Good morning. Thank you for coming. I'm Jérôme Boulanger from the Light Microscopy Facility. I will talk about image processing for light microscopy. So I think I will talk about will not directly apply to electron microscopy, but there are some overlap. But obviously the workflows are very different, and the problem as well. So the first thing is basically why do we process images? For example, we could want to improve the image quality. So we saw that we have a problem of number of photons, which is limited. So we want to improve the signal to noise ratio and correct background, improve reference frame to keep across images. So like image stabilization or image registration. We want to recognize, so this was the first part. We want to recognize object on features. So we want to attribute to a semantic class each pixel. So it's called semantic segmentation when you assign each pixel to a different class. We want to identify instance of objects. So distinguish between, so this was the first part, semantic segmentation. Then we want maybe to assign each block here to a different identity, at least above here. And then the next thing is we want to quantify what's in our image, what the biophysical process we have imaged on the microscope. So we want to measure fluorescence intensity, laser relative, or for example, some recovery time. We want to count number of cells, number of organelles, or foci in a cell. We want to determine or measure, characterize the shape, such as area, parameter, volume, roughness, or circularity, etc. Might want to measure dynamical information, quantifying motion, velocity, or amount of deformation. So what we want to do in the end is characterize a phenotype. So we have like a typical example, two conditions. We have acquired images for each kind of conditions, and we have our list of file. We can list the condition next to it, and for each file, we will, let's say, find the cell, find the nuclei, and measure the phenotype, quantify it, and assign some numbers. So it can be as simple as mean intensity, which will translate as an input, expression level, or some correlation between channels. And then in the end, what we would like to do is just be able to assign or quantify the effect of our condition or on our phenotype, which we are expressing here as a collocation for in Y type on the mutant. So this is the basic workflow. There are some other things, but roughly is what we want to do when we do image analysis from imaging. So what Nick talked about on Tuesday, to having a quantification, to use it as a tool to support our investigation. So how can we do this correctly, and how can we strive to make it working well? So we want to make it objective so that someone else will repeat the same analysis or workflow, will end up having the same conclusion. We want to be quantitative. So we want to, the measurements we are doing are related to our perturbation, our biological condition. We want it to be so reproducible. So again, a bit like being objective. So we want to be able to run again. And then we have, we want to be able to scale it. So often we want to quantify what's in an image, measure various quantities, but we want also to be able to scale it so that we have enough n numbers. But just before scaling it up, we often need to step a bit back. And I think this is a huge, advice I kind of learned, something I learned about the years, is that they are not just pretty picture, but we want to have number out of them. We want to quantify things, but we still have to look at them to get an idea of the data set. So our visual cortex is very good at understanding what's in the picture, it's what it's doing all the time. We can trust it to get an idea of, will this work? Is there information in my data? Is it not just clutter? So this is important to try to build an overview on your data set. And then, guess estimate, variability, try to have an idea of, can I hope to quantify something, to measure something from this? We need to discard or to explain outliers, looking at overexposed sample, that's et cetera. So in the end of the day, we don't blindly batch process thousands of images without a first rough look through the data set. And we need to have basically a link. Once we have our box plots, in the end, the final figure, a way to look at this outlier and try to trace it back to the image, to the cell, so that we understand why this point is an outlier, so we can eventually discard it because maybe it was saturated or a dead cell, or et cetera, et cetera. So it looks basic and simple, but sometimes because of the number of the quantity of data, which is 3D, which is maybe over time, and things like this, it might be difficult to do actually just this, trying to explain the measurement. How can we make it easier before doing anything? When you plan the experiment. So good advice is to add some housekeeping labels. So what we consider as housekeeping label is labeling a nuclei or cell membrane when we want to have information cell by cell. We want to control the cell confluence levels to avoid overcrowding environment where basically it will be very difficult to assign measurement from one cell to another one. We want to choose the right microscopy approach, so it's useless to have like a super resolution microscopy approach like Nick will talk next Tuesday to image just big average over the whole cell. So this is, you will waste your time, storage, my patience. But not my patience, I will just end up downscaling your image, or you will do the same. And basically it's just a bad practice. You optimize a bit your so basically do some first experiment where just to benchmark and to investigate what you can do, and try to and then when you do the picture, one thing which can help is to know what kind of analysis you do. You kind of think, okay, I take this picture now, but can I easily segment this cell? Can I count what I want to count just when you are on the screen on the microscope? This already helps you to ensure that will be smoothly analyzed afterwards. And then the last thing which can make it easier, sometimes an analysis task looks very difficult. You can't solve one little problem. At one step in the workflow, there is something very difficult. You don't manage to find a tool to do it. Actually, just doing it with a bit of interactive input will just solve the problem, and then you can move on for the parts which are difficult to do manually. So there is some parts which are just easier to do, a bit time consuming, but it will just smooth our workflow. So it can be like cropping the right part of the image or making a selection in the image which say, okay, I want to analyze just this part, and this simply will help you to have a simple workflow because you have focused the whole analysis on this part of the image. So what is an image? So an image, so we see images all the time on a cellular screen. So for us, there are measurements on a regularly sampled grid. It's not always, so it's regularly sampled. It can be a square grid, it could be an hexagonal grid, it could be a different kind of grid, but for us, most of the time it's a square grid. It can be like in 3D as well. So a volume, we can add time. So we see this as like basically an array or a big table of pixels. It can be also viewed as a function, a continuous function, which has been sampled at different locations. We can also go to the Fourier transform and see it as a sum of little wells. You can see it as a random field, like a magnet, to try to interpret the image as interacting potentials. You can see it as little wavelets or dictionary of feature of weight, and they are now in neural networks. So the basic thing we can do with an image is apply some filter, which is basically combining the intensity, the values in a neighborhood. We see, we use this device, this little square, which basically represents this neighborhood, or what you can call also a filter or kernel. And we will take all the values in this square and manipulate them using a weight, for example. Or if all the weights are the same, it will be an averaging, and then we will replace in a new image. We will store in the new image the value of the mean in the center pixel here. So this is called a convolution. We can also have some nonlinear filters where, for the same principle, we will, instead of computing an average, we compute a rank or like a median, a percentile of this little block. So, or the minimum or the maximum, and this is linked to this opening or closing or the dilation, erosion in our software. And another way of seeing this linear convolution is using Fourier coefficient. So we have a link between the convolution of the image on the multiplication in a Fourier domain. And yeah, so we can achieve various means by why we do this. So we can simplify the image by taking an average versus the noise level by the averaging. And then we could also sharpen the image to amplify edges, detect edges, or spots, features using simpler filters. Yeah. So, yeah, we can also have a different approach where we try to formalize what we want to do with the image, or the task we want to do. For example, so this is what we call Bayesian approach. So we have a task. We want, for example, to deblur the image, estimate motion, or denoise the image. So we have a link to the data. And since this link is not enough to recover the information, we also often add some a priori such as smoothness. And so it's what we have here. We have so a data fitting term, likelihood, here, and an a priori that we will add different cost function. We will compute gradients or an update, and we will minimize our cost function, providing a new estimate each time using our forward model to simulate an image and get an idea of the updated likelihood, comparing the estimate and the data. So this is typically what we have when we do deconvolution. We'll talk about it a bit later. And we can also reinterpret some simple filters in this context, where we could say a mean filter is also have an a priori that pixel in the neighborhood are the same. Another approach, which is often used, is learning-based approach, where we collect some input and output. So, for example, the input is the intensity at each pixel, and output is a label. And then we define a model which links the X and the Y. So the input in general will not just be the intensity, but the result of many filters. So compute like that filter, average, Gaussian filter at different scale, difference of ocean, and so on and so forth. So we collect for each pixel a vector, and we'll try to predict a label, which is just the class of the object. It could be also the whole image. The input could be the whole image, and the output could be a label. This is a kind of setup. Okay. In general, we still have often in the traditional what we call shallow learning, compute some features, so we don't use direct image, we compute some feature, and then we try to predict some label using a model. When we talk about deep learning often, we don't have this step of pre-computing features. We just let the training approach find the filter source in the most cool way of explaining. So again, we optimize the cost function using gradient descent or stochastic gradient descent, and then or another approach, which for example, SVM use different minimization function and also algorithm, and then we end up with tasks such as segmentation of object, detection of objects with eyes, or just classification of the whole image. So one task we talked about is improving the image. So when we do microscopy, also often have photon limited imaging. So the number of collected photons are a function of the exposure time and of the intensity of illumination. The photon interact with the sample, and so they create free radical on bleach the fluorescence. So we want to limit how much photons we can use to image. So in the end, there's sort of budget of photon you can use for imaging. And you can see here the effect of going from short exposure time to a longer exposure time. So the noise from just counting photons, they are Poisson distributed, so they are the strength of the noise, the variance of the noise is linked to the intensity. There is a dark current on camera-based system, so the photon generation of photoelectron, which is very low due to cooling. And then another important noise is the readout noise, which is just noise created by reading and count the electron and converting them into digital units. So you can characterize photoelectron or camera or sensor link the number of the gray value to the number of photoelectrons, so a quantification of how much photon were emitted by the sample or collected from the sample at each pixel. You can predict the noise distribution, and using all this information, you can compare sensor using this kind of number, like the readout noise and the QE here, which is how many photons reaching the sensor will be transformed into photoelectrons, and some other more noise-independent kind of features such as frame rate. So once we have noise, we can characterize it. We want to remove it to get an idea of what is the image without this this noise, which is a stochastic process. It's not nothing to do with the background, just stochastic. If we would have taken the image twice, you will have two, and take the difference, we will have only the noise, if nothing moved. So what we want to do is to remove the stochastic noise while preserving fine features and intensity. So we developed this method, which is using the whole 4D domain to do the denoising. So it works by comparing local, non-local patches in still an extended neighborhood in space and time. They will deal with Poisson and Gaussian noise, and so basically you exploit the 3D temporal redundancy opposed to virtually increase the exposure time. So here we have an example. Here are some more recent technique to do this kind of thing, such as CARE, for example, when you collect a pair of noise-free image and you train a neural network to predict a noise-free image from a noisy image. So here we have the problem of having to collect this image pair in advance. There have been an next iteration where you can use noisy image to train a network using the assumption that this is called noise to void, where you assume that removing the information from the center, one pixel in a neighborhood, and trying to recover it using training a network will enable to learn a denoising function. So we saw with Nick that our imaging was band limited. So when we said band limited, that there is a maximum amount of frequency. So this is what Abby found, a maximum frequency due to the light diffraction, which is here, our Fourier domain. So and this maximum frequency, which is collected by the optical system, is the order of the wavelength divided by twice the numerical. So again, this just that again that we have a like this sampling theorem. So if we have a regularly sampled band-limited signal at half the resolution, so twice the frequency, we can recover the signal exactly. So we have this sampling here. Yeah. So what does it mean for us? Is that if we have two samples here, one with little stripes in this direction and one with little stripe in this direction, takes a picture. So we don't take a picture with the frequency domain. This sample have these stripes appearing here and here. The optical system will be a low-pass filtering. So we cut out everything outside the cutoff frequency, and we end up with two identical images. So they basically lost the information about the orientation of these stripes. So this looks a bit artificial, but actually this is what happens when we go from here, so a sample image with a super resolution approach, where we see little actin rings along this axon. So this is storm imaging. And if we will take the same kind of image using, so here we see the ring, frequency here, we image with a normal conventional microscope, we cut up everything outside, and we end up with no rings. So we don't know if there is rings or if in which direction they are. We could ignore this. So we can't access this situation. So this speaks of notes toward the next presentation from Nick. So we can characterize in 3D the whole information of our optical system using a point spot function. We assume, when we do this, we assume that the response of the microscope is linear, so it doesn't change if we take 1 fluorophore, 2 fluorophore. The image will be the sum of the image of the 2 fluorophore. This is what linear means. And it's the same at each point of the image, so this is an assumption in general, but on the little area, there is no change of the response of the microscope, which is not true, especially now that the microscope start to have more and more field of view. So you can see point spot function of the object should be changing in the field of view. And so this point spot function is initially a function of the numerical capture of the wavelengths, and of course the sampling of your image, but also all the absorption. So you can capture everything about your microscope. Once you know it, you are tempted to try to take it into account. You know how to create a new image. So say if I have 1 fluorophore, now my image looks like a big blob. What about trying to convert to go back? This is what is called deconvolution. It will increase the sharpness because each big blob will be put back as a little point, and it will increase the signal to noise ratio because all the energy was spread over all many pixels will be assigned to one pixel. So we have an increase in resolution as well. The thing it doesn't do is doesn't denoise. Some people say denoise, but it doesn't denoise. It amplifies the noise because the main problem is that our optical transfer function, which is Fourier transform of the noise, which basically is this bright circle we have when I make the mask in Fourier space, goes to zero outside the optical transfer function, and then but the noise doesn't go to zero, so is end up being amplified. So in Fourier space, I briefly mentioned that the convolution was a multiplication. So the convolution we could do a division. Of course, we end up dividing by zero and amplifying the noise. So we can use a Bayesian approach. It's a noise model, and a priori to prevent this noise amplification to end up with reconstruction, which is sharp but not noisy. A common algorithm, the inner filter, HSMUC, or more advanced approach using particle gradient, for example. That's all the field on how to do this, invert these operators. So here we have access to various approach tools. So performing the convolution, you can use Huygens, which is installed in 1N120 and 2 PCs. You can use directly your metadata from the files. There's several algorithms, quick maximum likelihood, and good maximum likelihood. So it's that more Bayesian version. You can run on GPU, so pretty fast. We have also some deconvolution module for specific system. For example, ISIM needs deconvolution. The AirScan also needs deconvolution. And so we have access to this on the commercial software. We also have in Fiji deconvolution lab and a PSF generator. You can use OPS in Fiji. In MATLAB, there is UC deconvener. And in Python, you can also use Richardson directly from the scikit image, for example. It's just some pointer. There are many other ways of doing it. Next, talk about image registration. So image registration can be used when you have a shift between channels, for example, the chromatic shift. Just a motion, the dichroic, for example, used will shift a bit the image between two channels if you change using the turret, for example. So it will not come back exactly at the same position. Kind of drift. So your sample is moving over time, so you want to track it over time, or estimate the deformation or forces. Or you can want to time. So for tiling, you need to estimate the small error of motion when the stage went back to the same position. So you use overlapping pixels to adjust the timing. You can also register full samples to an atlas, for example. Register brains to an atlas, the brain atlas. For this, you can use either landmarks. So you have little points, feature of interest you have found, or you can set them yourself. You can use directly the intensity, or you can use a mix of the two. There are different kind of deformation models. You can have a parametric model where you say that the deformation at one point is a function of the position in the image. This position you can express with this, you can express translation. So basically, the deformation doesn't depend on the position, just to a constant vector in X and Y. And you can have rotation, rigid body, deformation, axial motion, etc., etc. So there are parametric models that can just be expressed as a function of the position. In the image, you have non-parametric motion, a dense deformation field. For each point, you can try to estimate a vector, which allows to register the two images. And this, for example, is used for motion estimation, trying to to follow fields in time. To perform this task, you can have knowledge-based approaches for global. So where you want just to estimate the global motions, so often parametric motion, using cross correlation. So you can find the shift in the image. Phase correlation. Yeah, so you can express correlation between two images for Fourier domain, or Fourier Balmer transform can also be used for rotation. We can also estimate the depth displacement field using correlation approach. So in it, it's a bit similar to convolution, except that we computed in little blocks the correlation between the two, between one block of pixel and a block of pixel in the next image. This approach is often very noisy, so there is no constraint between the flow or the field, the deformation field in one pixel is a deformation at the next pixel. And it's often called PIV, particular image velocimetry in several fields. Yeah. So another approach is non-map-based registration. So we try to find the correspondence between a point of interest and find transformation which maps the points. So often some point can be missing in one of the two images, so you use a robust strategy, ransack, or you can use also. So basically, you need to find two things: the correspondence and the deformation. So you try to estimate them in parallel. So now we have like a method called current point trees, which allows to do this. And then we estimate basically the parameter of the model using a least square. You can also have a non-parametric approach where you estimate the coefficient of a simplex spline. So we will not assume like a rotation or a parametric motion, but a more complicated deformation, which is parameterized with terms. So these techniques are available in PG with using BigQuant. In Python, MATLAB, you have PyCPD for point trace or in computer vision toolbox in MATLAB. In ITK, there is also various approaches for motion estimation. So this is a toolbox in C++, which is accessible from various scripting language for estimation of motion and segmentation of, in general, more medical image oriented, but very useful too. And finally, so we have different link between optical flow, particular image velocimetry. So when we have this dense motion, which we could, say, use block matching or correlation, local correlation approach to estimate its deformation. So we often assume the preservation of intensity over time. We estimate, so the point is to estimate the displacement of each pixel. We have ambiguity along contours, so we don't know all these stages. We don't know if they are going everything along a line like this is ambiguous. We don't know if it goes like this or like that. It gives the same result. So you need an a priori for smoothness to try to resolve these ambiguities. And often we use also a multi-scale approach for solving this kind of problem, trying to have a globally smooth solution at a low resolution and then propagate at lower resolutions. So once we have image deconfol register, we want maybe to find features of interest in our image. So we can perform some blob or spot detection. Often the object we want to localize or find the region just under the localization precision. It can be single molecules, physical, it can be on those ones. can be various things which appears on the image. Can be also a big thing, can be a nuclei, can be localized. It's a spot detection method. It looks like a big blobby thing. So you have this idea of multi-scale blob. So it applies to non-resolved object, but also as anything which we are not interested in a fine detail. Can be done using a simple local maxima. Often local maxima without pre-processing will not work. So you have some pre-processing, some blurring, some filtering before. You have the difference of Gaussian or Laplacian of Gaussian. So these filters are characterized by a scale. So basically, you use a certain scale, you will detect object at a certain scale. Some implementation allows you to have a multi-scale approach, which will give you the best scale of each pixel for a spot. So either specified scale, or because you know which scale to look at, or you can try to run the same filter for different scale and find the best response across the scale. So it's an easy way to detect physical or small objects, nuclei as well. It can be combined with model fitting, such as 2D or 3D Gaussian fitting, to have a sub-pixel localization of each of these blobs. So this ends up single molecule localization microscopy is based on this. It's easy to script either in any of the language or environment you want to use. It's also available in Imaris easily, or you can track them later on. And Fiji, you have also Thunderstorm, which is good for just doing some spot detection. You have the wavelet spot detector in IC. Yeah. So I talked about image segmentation at the beginning. So how can we do this? So again, I recall that the image segmentation is assigning a label or class to each pixel. So we have the semantic segmentation where we just assign a class, and instance segmentation where we identify identity. So this is cell 1, cell 2, cell 3. So we are able to separate the object even if they are touching. So the common approach is the simplest one, is just to threshold. So this will assign class 0 under the threshold is not what I want, above is what I want. This is the simplest way to do it. Once you have this, so this thresholding is often the last step of several pre-processing steps. You can enhance your image, remove background, you can do several steps and end up with a decision. You could have a multi-level thresholding, finding different class directly. You could have more complex methods for assigning class, so such as active contour, for example, some based method. You can have also some machine learning-based approaches, which will allow you to train the classifier based on examples. And then you have some deep learning methods. I just mentioned them just after. Once you have objects, a typical old-fashioned workflow would be to apply watershed to achieve instance segmentation using some seed, for example, which allows you to separate objects based on some either a priori or just the distance between to the contour. So all these methods are, you can find them everywhere. They're very accessible. You can do pixel classification using, so initially Weka in Fiji, for example, Elastic, and now you have also LabKit in Fiji. And then you can use some deep learning approach like QNet, Stardist, Self-Pose in Python or ImageNet for Stardist. I talk about Stardist. So Stardist is a method which has the deep learning method which has been pre-trained, so which makes it very useful for us because we don't have to go through the pain of annotation and training for especially segmenting nuclei. So I've been trained for segmenting nuclei images. They recommend each ROI we want to extract as a stack of X polygons, and we will try to predict at each point the distance to the contour. Like this, so the length of the radius here to the contour from the center to the contour, and the distance map. So it predicts these two things. And so we have here, so how does it do this? It takes the intensity of the image, applies some convolution, some downsampling, and then some upsampling using concatenation here of this level of convolution. So this architecture is called a unit. So it goes like a U like this, and the last level it will be having as many channels or channel as or output as a number of radius plus 1 for the distance here. So it's trained on a fixed predefined number of radiuses, and yeah, so the main thing is that it's very good at it has been trained, so it's very good to predict this kind of shapes which are convex. So that's the main limitation. It has to be convex. It works well in 2D. The 3D is a bit more difficult. So next method is cell pose. Cell pose has been used also a unit. So same, but you will try, the output will be different. It predicts the probability of being inside the cell. So probability of being inside the cell, here, inside outside, and the gradient of the distance function inside the cell. So by using this gradient, you can use a backtracking algorithm to find the center of the cell. So at each pixel in the image, we can backtrack if we are inside the cell, and all the points which will end up after backtracking in the same place will be assigned to the same cell. So this is, the network is the same, it's a unit. So the gradient tracking here, and from the gradient tracking, we end up having masks. That's been trained on many images of different types. And there is here the style part, which allows to detect during the training, during the prediction, the style of the image, and then directly use the right combination of weights to process the image. So it's very, so this to work well. So it works with two channels, ideally, one for the nuclei and one for something which marks the membrane or the inside the cytosol of the cell. So we have this kind of, so with these two channels, we'll be able to use a pre-trained network. They are also so there are several pre-trained networks for also just nuclei segmentation and also for membrane segmentation, actually. And yeah, it works in 2D and 3D. So the 3D is done by stitching the result applied for each plane in different directions. So it's not really 3D. It's not a 3D unit, but it's a 2D unit applied in each direction with some stitching steps behind. There is also, so this is what is pre-trained. There is also, you can find on the web some 3D cell pose version, but they are not trained on as many images or pre-trained. They are not as integrated. So next step is object tracking. So once we have found an object, maybe we have time lapse, we want to find identity over time. So that's what this tracking is. So there are often two difficulties or two facts which will make it difficult is the density in the feature space. Feature space can be as small position in X and Y. It can be also their apparent velocity. So if you move very fast, it will be hard from one image to the next to determine movement where simply. So To solve this problem, you can minimize an association cost, or a distance matrix, or distance between features space matrix. And you can also combine this with motion models to predict the next position. So it's called filtering. So what you can have in the term which could be used is a Kalman filter, for example, and there are more advanced filtering, but it's hard to they are not often implemented. So there are some challenges for tracking cells or tracking particles. So people try to develop techniques for solving this problem. So it's pretty hard. So the data is not to be immediately huge for cell tracking because you have large field of view, 3D all the time. That's just a lot of data. The go-to approach, which I would recommend, is TrackMate. So you use a different software for finding object spots, linear assignment for them. So that's cool. Lab tracker. It can also be combined with cell pools and star disk, or basically mask, but it's not so, or AOI. So there are versions where they will be able to process the output of some cell notation. There is nicely a very good spot tracker. Never managed to really use it, but it's very good performance. There is also a simple spot tracker, and simple, I mean simple to use. Yes, working well. It looks like the implementation is simple. It's very similar to the one in TrackMate, so in Imaris. And also Nikon elements also, which we have in the analysis room as some tracking features. Then once you have tracks, you can do some track analysis. You can be interested in instant velocity, measure variation of feature, for example, intensity over time. So the simplest thing you can do. You can be interested in mean-squared displacement to identify diffusion or sub-diffusion using, for example, here, multi-step model, or change between diffusion modeling on the, here would be like an active motion, like there is a microtubule or like molecular motor involved, and classify the trajectory over time, and also make maps of diffusion coefficient, something like this, using tracks. Another thing you can, once you have, here you can do without even too much pre-processing, colocalization, basically find the interaction between two proteins or labeling in your data. So you take an image with at least two channels, and you want to know, for example, here in yellow, it's appear in yellow because the red label and the green label are basically overlapping perfectly. But here, the red label and the green label are not matching, so it's hard to see any correlation. So, basic way to look at this is using this kind of plots where you have the red intensity versus the green in both cases, and you can see here they are aligned on the map. So this is typical of like a correlation between the two channels. So we can use a Pearson correlation coefficient to measure the amount of correlation. This is most basic intensity, no pre-processing, nothing, measurement. We can also use a it's called a Spearman, which is a bit more robust to non-linearity between the two channels. For example, if there is a change, it's not a line, but more a curve. You can use also, if you are segmented your image and you have some regions, you can use a Manders overlap coefficient using the intersection, the yellow over the red or the yellow over the green. You can, if you have localized spots, you can compute the pair correlation and the K function between the spots. So you compute the link between the radius to each particle. And basically, you count the number of particles around each particle in a growing ring. And then sometime, so you have just this kind of let's say one question, nothing to compare with, so you still want to know is the collocation measurement, the Pearson coefficient, the models, or the perturbation are due to randomness or blur or etc. So you can use some randomization tests for the Pearson coefficient. So basically, you scramble the image, you can recompute the same test several times. And here, when you have pair correlation and K-function, is a bit the same. You also have some simulation-based approach to determine if the curve you see the correction from here is real or not. There are also in IC like an explicit closed form formula for computing a p-value for the K-function, I think. Now, finally, we want to do what we'll call a hierarchical analysis, where you try to build a relationship between objects to assign using proximity of inclusion. So you have your image, in your image you have several cells, and in the cell you have several foci. You want to assign foci 1, cell 1, cell 1, and etc. And count, for example, the number of foci, or so you basically are starting to build a kind of semantic levels. You have image or count of an organ, a cell, some organelles, and you can use math and logical tests to get the index of the arrays or compute distance transform to know if foci 1 is like cell 1 or into a nuclei 1, etc. So this kind of approach of thing is very easy in GS3, so general Z3 in the Nikon software. In Fiji, you will need to manipulate their eye to find if they are intersecting or not. And there is a separate module in Maris which allows to build this. Just a summary of software and tools. So the go-to thing, because you have access to it on your, it's really easy to install, is Fiji. So you can open your image, look at it, apply some first existing tools, and just stop there. It works. You can use also Imaris, which is very good for 3D visualization, creating a rendering also if you want to impress your friends or colleagues. It's a nice 3D rendering. That's very good. And also everything is detection and tracking, and there's also a localization module. For GS3, I would recommend this for simple workflow. You know, that's a simple approach with scaffolding on region manipulation, and where you have a lot of data, but simple approach, and your images are very similar to each other, so there is no big change. Otherwise, the workflow is a bit hard to customize and to adjust. Very good for getting started and getting also an idea for how to organize the data. Against, so this is another commercial software which we can use for deconvolution and collocation. So it's basically a state of the art still. Napari. So this is a tool for visualization which is written in Python, but we just install it and open the image without having to do anything in Python. And using plugins, you could achieve something similar to Imaris. There is Appear, which is a Zeiss online workflow. So everything is online. You upload your image on a workspace, and then you can process them using like non-programming graphical interface. Cell profilers also for training workflow, a bit in the same logic on GS3. IC is a general tool for spot detection, tracking, and collocation. And there is scripting approaches here. One more minute, just to say that okay, we are here to help. So at the moment we are two. There's Dina, the back of the room, and myself. You can come and talk to us if you have if you want advice, or if you want help on like scripting or anything related to image analysis. And we here, my list will be short, but we have a list of training on various. So I try to revamp it and we make like a plan for the year, but still not yet there. But I try to provide you more in-depth training for around imaging. We have one in Python soon, so we did it last year. Dina did one on organs. We will try to redo one on GA3 and polymaths. Thank you for coming.